Hi, my name is Alexandra Alexiev, and I'm a PhD student here at University of Colorado Boulder in the lab of Valerie McKenzie. I'm here to tell you about the summer work that I've done that's been supported by the Indian Peaks Wilderness Association and where it's going in the future.
My work is part of the McKenzie Lab's mission to study the locally endangered Colorado boreal toad and their skin microbiome. Colorado has just this one high elevation toad species, and it lives in shallow beaver ponds such as this one, which we sampled this summer. Amphibians are interesting from a microbial perspective because they go through a dynamic life cycle where their body goes through drastic physical changes as they go from an aquatic to a more terrestrial habitat. All these life stages are a really integral part of the food web system at high elevation ponds. Unfortunately, our Colorado boreal toad is threatened by a globally devastating fungal pathogen called BD. BD infects about 40% of all amphibian species across the whole planet and is highly fatal to many of its hosts. Fortunately, the McKenzie Lab and other labs around the world have found strains of microbes that live on amphibian skin naturally that can combat BD in the lab. In Colorado boreal toads, we know a lot about the bacteria that might be able to combat BD, but very little about the fungi. To characterize the role that fungi might play on our toads, we asked 3 questions. What is the diversity of culturable fungi on toad skin? Which of these compete against other microbes in culture, and what metabolites lead to this inhibition? To answer our questions, we sampled at a small pond in South Cottonwood in Chaffee County. We found toads at all the life stages, but here are some pictures of just some of the individuals we sampled. For each animal, we swabbed its entire body and then rubbed those swabs on fungal media plates. The plates provide each fungal cell on the swab with nutrients so that they can grow enough for us to see them and then eventually identify them. We visited this pond 5 times throughout the summer to catch all the different life stages. After letting the plates grow for a while in the lab, we grabbed all the visually unique fungi from each plate. We initially predicted about 100 or less different fungi, but have harvested over 150 from our plates. This is incredibly exciting and gives us a clue that toad skin is actually very rich in fungi, which is one of our main questions in this study. We use these super diverse plates to isolate the individual visually distinct fungi that you see here into pure culture, which is necessary in order to identify them. It actually took us all summer to get all of these into pure culture, especially since many of them grow incredibly slowly. For example, it might take a few weeks in order to get this much growth. Here are some examples of the fungi that we've found so far. We have a number of different aspergillus isolates, and this group is actually really common in most environments, but we found it especially enriched on our adults and subadults. You may have heard of this fungus as something that can make humans sick, but we actually don't know yet the exact type of aspergillus we found, and it is very likely that this doesn't make humans sick. We also found a few fungi from the group Penicillium, and you may recognize this group's name since it includes the fungus that originally produced penicillin, the first commercial antibiotic. This group includes a lot of other types of fungi though, and we found one of them on our toads. We hope that this one might be able to fight off BD in culture experiments, which are the next step in our work. Another interesting fungus we found is this Fusarium. This group of fungi are common in most soils, which makes sense since we found it on our adult toads, and adult toads hang out and burrow in soil. The one shown here probably isn't dangerous, but there are others in the Fusarium group that can cause plant disease sometimes. And our eggs had lots of this pink pigmented yeast called Rhodotorula, and this group has been found in a lot of places like air, water, and soil and dairy. Some yeasts can produce antibiotics, and so we're also interested to see if this one is going to be able to fight off BD as well. Right now, we're working to extract the DNA from these fungi since we need to know what they are before we can compete them against our bacteria and BD. So we put the fungi into these tubes and we use a machine that sucks out the liquid from the fungal tissue. Then we use this little pestle in order to crush the fungal cells. And this is because fungi have very tough cells and we want to get as much of the DNA out as possible.
So before they go into this machine, they look incredibly wet and soggy, much like this one. And when they come out of the machine 24 hours later, they're much crunchier and drier, much like this one. Once we know who we have, we'll grow them on the same plate with one of these 4 bacteria, or BD. The bacteria we've chosen are common and dominant on toad skin, and we know this from past research. We want to make sure that we know not only how toad fungi interact with that BD pathogen, but also how they interact with some of the bacteria present. In particular, we want to know how they interact with some bacteria that we use as probiotics, like that one. And we want to make sure that bacteria like this one will not be killing off naturally occurring fungi on toad skin in addition to the BD, or we might find a fungus that can help that bacteria stick for a longer time on toad skin. The difficulty with most probiotics is that we don't know the full extent to which all of these interact with each other, and if we want effective probiotics, we need to take the whole community into account. I hope you've all enjoyed hearing about the work that you've supported, and we really appreciate this grant. It's definitely been a long process this summer, but we're really excited based on what we've isolated so far. And we think we'll be able to uncover some really cool microbial interactions that might benefit the Colorado boreal toad.